I'm Gordon McVie, I'm Managing Editor of eCancer, and I'm highlighting in particular this month a couple of papers from Pan Pansiarka and his colleagues. They're involved in a project which is described in one of the articles, the ReDO project, and it's all about repurposing established drugs, finding drugs which have been around not necessarily in the oncology world, but in the formulary, and testing them for anti-cancer effects. The advantage of these drugs, the obvious group are the anthelmintics, but there are others as well, such as trinitrin and cimetidine. They're off patent, so they are very cheap. They've been used in thousands and thousands and thousands of patients. The toxicity profile is low, so they're highly applicable for use in developing countries. The ReDO project is funded by the Anti-Cancer Fund from Belgium, and the project is setting out to campaign, in a sense, for doctors to accept the fact that the present-day breakthroughs, which are described most weeks in eCancer, are really only going to be affordable for Western civilizations, those with money, and that we need to tackle this immense problem of cancer, which is developing in our developing countries. They have two approaches. One is to look at established drugs, as I mentioned, and to retest them, and in part perhaps to redesign them for use either as single agents, but they argue strongly that they would be used as adjuvants and as additive drugs to established acceptable drugs which are proven to be effective. And the second approach is to take low dose, continuous dosage use of established oral drugs which are low in cost, and they are taken by mouth, and so will cut down the costs of going to hospitals and having injections. The metronomic therapy group, the advocates of this group, met in the IEO in Milan a couple of weeks ago, and eCancer covered the project. Pan takes one drug in a separate article and discusses why it deserves a new look. Mebendazole is a well-known anti-worm agent. It works against most worms, little threadworms and tapeworms, and more aggressive and nasty, life-threatening worms and other beasts like echinococcoses. And it's got a very low toxicity profile. It's been used for many, many years, and it is effective in large numbers of people who suffer from mostly gastrointestinal worms. Evidence that this drug, mebendazole, and several other in the class of drugs has anti-cancer activity. It's been around for quite a long time. It's been ignored because it's not a classical drug. It wasn't clear what its mechanism of action might be, but that's all unraveling now, and this paper shows that this drug and some of its analogues are actually active in vitro in lung cancer cell lines, in vivo in xenografts, in adrenocortical tumors, in breast cancer models, active in colorectal models and in lung models. And the enthusiasm for following this approach was stimulated by a couple of case reports which are described in the article. One is a patient who had adrenocortical tumor which had been treated unsuccessfully, as is nearly always the case, by a number of anti-cancer drugs, and the patient's decline was halted, and there was a progression-free interval after taking this anthelmintic of over a year. The second report is really quite spectacular. It's a patient who's advanced colorectal cancer who'd had all the usual candidates, the fluoropyrimidine, irinotecan, antibodies, and had progressed through them all. This patient had measurable lung metastases and was given a course of oral mebendazole, 100 milligrams twice a day, and underwent an almost complete remission as measured on sequential x-rays of the lung. This response lasted for well over a year. And it's of considerable interest that this patient was taking no other medications. So one can't really escape the conclusion that this drug did produce this anti-cancer effect. So what? Well, ReDO, that's the project, are saying, well, what about other drugs of the same class? They're not well known for their absorption from the gut, but there are now liposomal preparations available. What about looking at those? There is a possibility for slow release mechanisms and formulations, which might be even more useful to be tested, A, against more xenografts, but B, in phase 1, phase 2 clinical trials. Either solo agents, but the ReDO group emphasize as combinations with other active drugs. So I commend this approach to you. eCancer's in behind it because we campaign too on a variety of issues, and we are passionate about the search for solutions for better cancer care for cancer patients in developing countries. eCancer is a global journal and is read by oncologists in 192 countries, I think the last count was, 40,000 visitors per month. So we can make an impact, and I hope you'll read these two papers and think about them and maybe do something with the data.